Okay. It is echoing. Shall I join again?
Just a second, sir.
Sure.
Good morning. Good morning, everyone. A warm and hearty welcome to one and all present here for the inaugural function of MSAPTI sponsored e-National Conference on Metabolomics, hosted virtually on 25th and 26th of April 2025. It gives me immense pleasure to be your host for today's inaugural session. I'm Professor Manisha Puranik, and I feel honored to welcome you all on behalf of the organizing committee. This conference aims to bring together leading researchers, academicians, scientists, and scholars to share their valuable insights and research in the emerging field of metabolomics, a field that holds tremendous potential for breakthroughs in healthcare, diagnostics, and drug discovery. Let's begin this beautiful academic journey by acknowledging and welcoming Principal IPER. We welcome Dr. Rajendra Ganjiwale sir, Principal IPER, and all the respected faculty members, participants, and students who have joined us from across the nation. Traditionally, we light the lamp to symbolize the dispelling of darkness and the awakening of knowledge. Though we are connecting virtually, let's symbolically light the lamp of wisdom and enlightenment and let us invoke the blessings of the Almighty.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
Now let's take the aerial view of IPER Wardha. Let's take the aerial view of IPER Wardha. Thank you, everyone. This is our beautiful green campus, IPER. Now I take this opportunity to invite Dr. Rajendra Ganjiwale sir, Principal IPER, to formally welcome the gathering and give an introductory address. Dr. Rajendra Ganjiwale sir.
Good morning, one and all. On behalf of the IPER, Wardha, I heartily welcome respected dignitaries, esteemed guests, distinguished speakers, dear faculty members, students, and scholars to this inaugural ceremony of APTI-sponsored virtual national conference on metabolomics. As you know, this metabolomics, it is one of the recent approach in the pharmaceutical sciences, which deals with the study of the metabolites which are being produced as the result of the process of metabolism within the cells, tissue, or organism. Metabolomics involve quantification and identification of the metabolites, and it's a direct correlation with the disease because of the imbalance of the metabolic level that will indicate the abnormalities and the changes in the body. In today's online conference, we have invited the renowned speakers, resources, and experts of the national and the international repute. Among them include Dr. Shyam Kumar Maskapalli, Dean and a Professor, School of Bioscience and Bioengineering, IIT Mandi, Himachal Pradesh. Welcome, sir. Dr. Kanaya Kumar, Senior Scientist, Fermentation and Microbial Biotechnology, CSIR, Indian Institute of Technology. Then Dr. Dada Saheb Kokre, Professor in Pharmacology, Department of Pharmaceutical Sciences, Nagpur University, Nagpur. And my esteemed colleague, Dr. Syed Ahmad, Director, Center of Excellence in Unani Medicine, Professor in Department of Pharmacognosy and Phytochemistry, HOD Food Technology, Jamia Hamdard, New Delhi. Welcome, sir. At this moment, I'd like to take this opportunity to express my heartfelt gratitude to the Maharashtra State APTI for sponsoring this prestigious conference. I am also deeply thankful and I'm going to express my sincere thanks to my chairman, sir, who is the main custodian of this institute, which has always stood as a pillar and a strength and guidance. Although he is not physically present with us, but we are truly honored to acknowledge the support and the vision of the president of the Custodian Society, Honorable Dr. Nitinji Dhande, sir, in his absentia. A special thanks to the all keynote speakers whose presence today increased the academic value of this conference manifold. Your insights, expertise, and deeply appreciated. We eagerly look forward to your address. I am also thankful to all our plenary session speakers, eminent scholars, researchers, whose participation adds a tremendous value to this conference. Your contribution will be undoubtedly enlighten our audience and open a new dimensions for academic and research pursuit. Lastly, I wish to thank to all participants, organizing committee members, supporting staff who have worked diligently behind the scene to bring this conference to the fruition. Let us make the most of this opportunity to learn, share, and collaborate. With this, I hereby declare that the today's Maharashtra State sponsored APTI conference, e-national conference on metabolomics is officially inaugurated. Thank you all. Wishing a productive and enriching conference ahead. Enjoy the deliberation. Thank you. Thank you very much.
Thank you, sir. We now proceed Thank you, sir. We now proceed to the release of the conference e-proceedings, which contains a collection of speakers' talk messages of this event.
Hello, thank you very much.
Thank you, everyone, for being part of this symbolic release of the proceedings of e-conference. As we draw the inaugural session to a close, I invite Dr. Dilesh Singhvi, sir, IQAC coordinator, to propose the vote of thanks and acknowledge all those who made this event possible. With that, we officially declare the MS APTI sponsored e-national conference on metabolomics open. Thank you everyone for joining us to inaugural session of e-national conference. Over the next 2 days, let's engage, explore, and enrich ourselves through insightful sessions and stimulating discussions. We request all the participants to join the next technical session as per the conference schedule, the link of which is provided in your WhatsApp group. Thank you once again for your presence and wishing you all a productive and enlightening conference ahead. वो connect Sir, Shyam Kumar Masakapalli, sir.
Yeah, very good morning. Am I
Sir,
audible?
you are co-host now.
Okay. So that
And
would mean I can share my screen.
Oh, I'll start with your session, sir. First, I'll give a brief introduction and then you can proceed further, sir.
Sure. Can I share my first slide? I mean, let us check.
Sure, sir. No problem. You can share your first slide, sir. The title slide of your presentation. You can share, sir.
I'll share my entire screen so that.
Yes, sir.
So can you just confirm if My screen is visible completely.
Yeah, your screen is visible, sir. Completely it's visible.
Thank you very much.
So I'll give just a brief introduction and then the virtual platform is yours to present.
Okay, thank you.
Okay, sir. Good morning, respected speaker and all participants joining us from across the country. I'm Dr. Manisha Puranik, and I'm honored to be your moderator for this plenary session at the E-National Conference on Metabolomics, hosted by Institute of Pharmaceutical Education and Research, Wardha. Metabolomics, as we know, lies at the heart of understanding systems biology, revealing insights from plant sciences to human health. Today's session brings together leading minds to share frontier research, innovative methodologies, and real world applications shaping this transformative field. Our first distinguished speaker is Dr. Shyam Kumar Masakapalli, sir, Dean and Professor, School of Biosciences and Bioengineering, Indian Institute of Technology, Mandi, Himachal Pradesh, India. His core expertise is in the areas of metabolic systems, biology of plants, and microbes, with focus on metabolism, metabolomics, 13C fluxomics, phytochemistry, cellular bioprocessing, and agrotechnologies. He is serving as the board of editor of ASM Journal Systems. He supervised PhD students, postdoc students, MTech students, BTech scholars at Metabolic System Biology Lab of IIT Mandi. He served as a founding chairperson of School of Biosciences and Bioengineering. He also served as a course coordinator of MTech Biotechnology and faculty advisor of BTech MTech Bioengineering. He received Distinguished Teacher Award at IIT Mandi and executed 16 prestigious projects funded by ECR SERP, DBT BMBF, Imprint, Spark, DBT, Farmer Zone, and DST, etc. Dr. Masakapalli sir received PhD in plant sciences from the University of Oxford in 2012. He worked as a postdoctoral fellow first at University of Oxford and later at University of Bath, UK. He joined IIT Mandi in 2015. Thank you, sir, for joining us today.
Thank you very much, Dr. Manisha. It's really a pleasure to be here. Also,
So now the virtual platform is yours to deliver the lecture, sir.
Okay, so I hope I'm audible to everybody.
Yeah, you are audible, sir. You are audible and the screen is visible also.
Okay, thank you very much, Dr. Manisha. It's really a great pleasure, and also the platform. Thanks for providing that. I will try my best to complete it in about 40 to 45 minutes, followed by if there are any questions, I'll be very happy to take. So what I would try to do, as the theme is metabolomics largely, I would try to go and share real world problems on which we work, which encompass metabolomics as well as other associated technologies. And largely, given that the institute is pharma related, I would like to link it with the therapeutic bioactive molecules which we discover from the nature. And also I will touch upon some functional foods, R&D, which we are actually pursuing at the moment. So just as I've seen the beautiful campus of your institute, I would also like to show where we are based. One of the northernmost IIT, of course, just below IIT Jammu, but you can see IIT Mandi in the Himachal Pradesh. This is where it is located. And the pristine campus, as you can see, is nestled within the beautiful terrains of mid-Himalayan region. And we do have two campuses, North and South campus. And you can see here the North campus. This is where largely our engineering as well as research teaching programs are operated. We do have the South campus, which is again largely the basic sciences as well as research, teaching, student hostels, and so many programs are being operated here. We also have high-end facilities here. Among these, one building is the School of Biosciences and Bioengineering. Of course, it's spread over 2 buildings right now. And School of Biosciences and Bioengineering, that is where I am located and based, and my research group works here. As you can see, we are in one of the most beautiful campuses in India. Apart from enjoying the nature, we also do some research. I wanted to really share the sort of research my group in particular does. So my group works on largely metabolic systems biology, and we are focusing on both the plant systems as well as microbial systems, wherein we wanted to understand their metabolism, intracellular metabolism, the pathways which uh, uh, really help these organisms to overcome any stresses, or even in their natural conditions, how they really survive. Apart from that, we work on plant microbial metabolic crosstalk, because that's one of the very important areas, having immense roles in agriculture as well. So we do a little bit of work on smart farming, and today I'll touch upon some indoor farming based functional foods which have got therapeutic potential. And also I'll speak about the phytochemicals from the Himalayan diversity. So along with the research, we also started since last 7-8 years, we have been working on profiling these nature small molecules or the phytochemicals from the Himalayan region, and we wanted to see if these could serve as a repertoire of any novel microbes, novel molecules, or phytochemicals with therapeutic potential, and also other range of applications. I will touch upon this. Largely, today's talk is focused on this phytochemicals and the therapeutic potential. Apart from that, we also work on some waste to value projects. Now, these are the facilities and prototypes I'm just sharing so that any scholars wanted to visit us or come and spend some time with us here at IIT Mandi. My group is supported with analytical platforms. We do have common laboratories, plant biology lab, as well as low-cost polyhouses and so on. Apart from that, we also collect samples from the nature in order to do our profiling. So I'll share some of those. In a nutshell, today's talk will highlight the tools which will help us in accelerating the discovery of these novel therapeutic molecules. That will encompass metabolite profiling, small molecule analysis, modeling, simulations, experiments, and also I'll touch upon the thematic area of metabolomics. Then how metabolomics is helping us to address some of the real world case studies will be discussed. In the process, I will also present a novel Himalayan medicinal and aromatic plants and phytochemical database, which can help many researchers in India and abroad in the coming era. Okay, and at least 3 case studies I'm going to share, largely on antiviral therapeutics, how these small molecules help us, antibacterial therapeutics, the sort of work we are carrying at the moment, and also some of the R&D based on which we define some of the greens as functional foods, or you can call it as nutri-metabolomics based approach to define the functional foods for therapeutics. So just to give a brief introduction about metabolomics, all this, any biological system you take, they of course take the carbon sources, available nutrients, minerals and others, but eventually the enzymes within these cellular systems, they help convert one molecule into another. These molecules which are part of this metabolism, which participate in metabolism, are metabolites. These are largely divided into like primary and secondary based on their roles, their functions. Primary is essential pretty much. Secondary are very specialized. Whenever the plant wants, they really express it to protect themselves from various stresses, whether it is biotic or abiotic stresses. Inherently, also many plants produce their own unique secondary metabolites, which gives an identity to them. Like tulsi has got its own aroma and other stuff. So in a typical plant, you see it can be divided into many organs: leaf, flower, stem, root, and then and they are also continuously under biotic or abiotic stress. Now, if one collects these samples and wanted to ask a question about what is it composed of, what molecules is it composed of, especially at the level of metabolites, then for that there are various analytical techniques. Thanks to all the physicists, chemists, engineers who developed these techniques, analytical platforms like mass spectroscopy, HPLC, NMR, nuclear magnetic resonance spectroscopy, or even separation, all the chromatographic techniques. So these will help us to really look into like central carbon metabolites, phytohormones, flavonoids, phenolics, terpenoids. I will touch upon these, which are many of them are therapeutic molecules, and other molecules like aromatics, volatiles, and so on. So, but these are not isolated inside a system. They are a part of a bigger metabolic network. So where one can really look into their relationships, their associations, how they contribute in various pathway activities. So, but as a snapshot, at any time point in an organ, if I click a photograph, especially the photograph of all the metabolites, I call that as metabolome. The set of all the metabolites present in a cell is metabolome at that particular time point. So if I do across the time points, we call them as kinetic metabolome, or you can say kinetic metabolite profiles. Okay, so when we say about omics in the context of measuring all the metabolites, and you wanted to really compare between various time points, or compare between different organs, or compare between different varieties, in that comparison encompasses multivariate statistical analysis. Number one, you may use analytical techniques to measure them. Number two, you may use statistical methods, or if you look into some of the papers, they may mention it as chemometric analysis, like principal component analysis or partial least square discriminant analysis. So these kind of multivariate statistical analysis will help us to see how different are the samples. So that provides us, gives us an insight about the number one, identity per se of a system, and how different is it between various systems or various cellular systems. I'm going to touch upon those. Typically, this is your sample. Once we prepare the sample using any of the solvents, whether it could be a water-based solvent, or inorganic or organic-based solvents, largely you take like methanol, ethanol, ether. You extract the molecules by grinding traditional methods. There are a lot of protocols. I would encourage the scholars listening can find metabolomics workflows. Then they can extract the samples, and once from there it starts, where the analytical platforms will help us to measure what are the molecules present in them. And am I audible? Sorry.
Yes, sir, you are audible.
Okay.
Yes, sir, you're audible.
Yeah. So then there is a lot of series of steps. Once you get the spectra or what are present, then this will act like a fingerprint. From there are a lot of steps, like say alignment of the spectra, peak identifications, deconvolutions, followed by statistical analysis, compound identification against existing libraries based on either elution times or based on their masses. There are a lot of ways by which one can do, and these protocols are pretty much available. One can really learn this. A lot of online lectures are also freely available to really get some hands-on insights into these particular knowledge. Finally, one can use this data in order to make the interpretations. So the case studies will give some clarity about how we can really do the interpretation. So before going that, why are we really very motivated about plants in particular? Is because they are the nature's doctor, in a way that they have so many bioactive phytochemicals present in them. Due to photosynthesis, the carbon is driven into various small molecules. The small molecules, they combine together to make complex molecules, like whether they go via shikimate pathway, mevalonate pathway, or alkaloid biosynthesis, and you see all this range of small molecules. They can be largely categorized as phenolics, terpenoids, alkaloids. For example, let me give you a few examples here. You see, Nobel laureate, is awarded Nobel Prize for her discovery of plant-based therapy against malaria. And the molecule on which, based on her work, which was largely studied, is artemisinin. and it's an anti-malarial compound. This is a source from the plant, Taxol. Even now, it is used very widely in chemotherapy and others. That's like Taxol, or there are others like, say, vincristine. You might have studied these, have got very therapeutic applications. So apart from that, there are several which acts like analgesics. They can act as like cardiotonics. Again, so many of the molecules are there. So today in our talk, we will hit upon some molecules which have got potential as antivirals. Given the pandemic and our own experiences, I'm going to share some of our experiences. And also I'll look into some of the challenging, how these phytochemicals and other small molecules can also act as antibacterials, especially looking into the challenges of MDR, multi-drug resistance, which is largely being experienced these days. Many strains are getting resistant to our existing antibiotics. So we need to come with new molecules. So I'll share some of the progress which we made in both the directions. So before that, I will focus on the profiling part. So Himalayas, given that we are located here, they house a large number of medicinal and aromatic plants. In fact, there are about 22% of medicinal plant species which are confined to the Himalayan terrain. These are very much endemic, which means they are very much localized, and you don't see those kind of plants elsewhere. And India, as a whole, you see 44% of medicinal plant species you'll find in India. Large numbers we do have. Compared to other countries, you can see India is well placed to house a lot of these small molecules and medicinal plants. Now, given that IIT Mandi is located in this pristine Himalayan terrain, we thoroughly looked into the sort of plants which are present here. And we understood that there are several threatened medicinal plants. About 58 are somehow mentioned in the literature, as well as by some of the organizations. And also there are several aromatic plants largely used in industries, and some medicinal flowers, like, say, rhododendron, and both the varieties. It happens to be either the state flower or state tree of Uttarakhand, Himachal, Nepal. So these we wanted to study this. So what we did, we started exploring this Himalayan flora, and we wanted to tap them for drug discovery. So that is our goal. In relation to that, some of the relevant case studies which we will be discussing is, number one, we'll look into the Himalayan medicinal and aromatic plants, greater than 50 threatened medicinal plants. And we'll see how we do the profiling, which will help us in doing the metabolic analysis or metabolite profiling, leading to the metabolomics and also drug discovery. And all the information what we get, how we developed a Himalayan specific medicinal and aromatic plant phytochemical database. So that is a very rich resource of these small molecules. And now this database can serve as a repository from where we can take the molecules or the plant extracts and try to really hunt for their activities against viruses or against bacteria, or even as functional foods. I'll touch upon some of those case studies which could be very relevant in today's lecture. Now, the first case study is comprehensive phytochemical profiling of selected medicinal and aromatic plants. And we use multi-analytical platforms. Some of the examples are like rhododendron, and also essential oils from Himalayan aromatic plants. Let us start with the rhododendron red and pink varieties. If you were to ask a question, okay, somebody is making juice out of this or a squash out of this and drinking, what are we actually subjecting our body to? What molecules are we really being exposed to? You can ask a question. In order to understand that, the first point of action one needs to take is carry the metabolite profiling. So you do the hot water extract, and the metabolite profiling can be done using, like, say, analytical platforms like proton NMR, liquid chromatography, mass spectroscopy, or gas chromatography, mass spectroscopy. So you can spectrometer. You can use these these analytical platforms, and you can see the end result of this extract is some sort of spectra. If I don't label the spectra and ask you to try to label, you would actually try to go to either, if you have a standards, you would like to compare against them, or you would go to a library and really search for the library based on various factors, like, say, their masses, their elution times, or their what we call the ppms or the splits in the nmr, so we take all these information and eventually we assign these molecules. And interestingly, we find these flowers are rich in quinic acid derivatives. So, for example, they have got like caffeoylquinic acid or chlorogenic acid, as well as there is like coumarylquinic acid, there is catechin, there is like protocatechoic acid, and the amounts of these quinic acids, you may argue there might be other plants where quinic acid and derivatives would be there. Yeah, but the the amounts in them is very less compared to these flowers. So, and since centuries, people have been using this. But before even trying to look for any applications, we thought let us compare using metabolomics approach between the pink and the red variety. Red bar for the rhododendron arboreum, red colored flower, pink for the pink colored flower, which is rhododendron campanulatum. So using this multivariate statistical analysis of those peak intensities which we got, we could see that there are indeed differences among them. And although there are a lot of common metabolites, their relative abundances could be different. So that means their bioactivity levels, they could be different. If each flower could be actually fine-tuned to a specific application, or if one wanted to really extract these phytochemicals and come out with some formulations, it is really possible. These studies will help us. Now, let me briefly try to map the small molecules, how these are being produced in the flowers. If you look into the shikimic acid pathway is one of the key routes by which these quinic acids are produced. Okay, or it will go further downstream, and then you will see that a lot of this coumarylquinic acids, caffeoylquinic acids, or maybe catechins or epigallocatechins, the flavonoid biosynthesis, these all are being produced in this particular flower. So again, the pink or red, the presence or absence is being mapped here onto a pathway. Similarly, if I go with the essential oils, about 28 essential oils we profiled, largely used in industries, including our soaps, in perfumes, in several medicinal formulations, these oil extracts are used. So as an example, what we do, we take a typical oil from the raw material, the oil is extracted. We do have industries here who extract it for us. And we collect those samples, bring those in purest form, without any contaminations and other stuff. And we run again the profiling experiments using multi-analytical platforms. And then we try to understand what are present in those. And we could see which are the most abundant metabolites. So now these will help us, these metabolites, their structures, their functions, one can really predict those. And what we ended up is, for this 28 aromatic plants and the essential oils, we ended up with something like 2,500 phytochemicals, out of which at least 600 are very unique across all the essential oils. That means they are present in only in one or two essential oils. There would be common molecules also, but largely there are also unique metabolites. And 90% of them are terpenoids. Now, we figured out these terpenoids, many of them are have got their own functions reported as therapeutic molecules. And we see that many can be like monoterpenes as C10 molecules, very complex. Generally, these are derived from isoprene units like C5 are isoprene units. Then they combine and make monoterpenes. Sesquiterpenes is C15, C20, or C30. So what then we started understanding, okay, this is the chemistry of these largely these terpenoids as well as other phytochemicals. Can we really map them in such a way that one can really readily find the right source of plant in case they find a small molecule which is acting against a drug? So we wanted to map them onto a pathways. So that is how we did here. I represent here, simplify for about 10 essential oil components. The color coding means the molecules are present or absent. For example, let me just take one example. Let us say ocimine. So this color, it must be present in, say, tagatus, or maybe I'll take the valeriana. Valeriana has got camphene. Valeriana has got borneal acetate. So that is the way how I can really map them. Now, if somebody asks me, okay, this particular oil, what molecules are present, I can quickly color code and have a look into this information. So we created a database which would be accessible to everybody very soon, and that would help us to really look for various applications, as I'll show you how we do the drug discovery from this. These are like all these complex molecules. You see, the plants have very wide repertoire of molecules, and who knows, one of these molecules could be the molecule which would solve a health crisis altogether. And so, if we are lucky enough, we may hunt and we'll find their bioactivities. So major takeaways from this first case study is that multi-analytical platforms will help in phytochemical profiling. I'm sure every state has its own phytogeographic region, their own plants, their own traditional knowledge. If you can really tap into them, you can come to the scientific basis of what molecules are present in them and whether they are responsible for any bioactivities. We deciphered about several phytochemicals from 2 rhododendron species, the flowers, and also 28 essential oils. The metabolomics and pathway analysis captured the rich phytochemical diversity, as you've seen a lot of molecules down there. So for further studies, you can refer to our work, which are recently published. The scholar also has completed her PhD. Shagun, I would acknowledge her. Now I'll move on to the second one. So given the information is already captured, now the question asks, is there any way of making it accessible to everybody? The best way is to really create a database. So we thought, why don't we develop a HMAP square database, which is Himalayan medicinal and aromatic plants and phytochemical database. So we took about 50 medicinal plants and we confined ourselves to the threatened, rare and endangered medicinal plants. We call them as red species, and few other plants also, which we feel like they could add value, and essential oils, about 28. Other plants are these flowers. Actually, they are not threatened as at this moment, but we included them. And in this website, you may find, if you log in right now, it may have password protected, but if anybody is very keen to really experience it, you can write back to me. I can provide you the password and the user for your own use until we release it in the open source. Now, these medicinal plants, for example, take the case of aconitum, podophyllum, some of those plants. I'm not going to get into the details of that, but I'll come to the main takeaway. So from these medicinal plants, if you could using various extracts and plant parts, if you find these are the molecules which are present in them, those can be mapped into the library, into the database. So what we ended up is about more than 2,500, if I add all of them, more than 2,500 phytochemicals we deciphered. And the spectra are available. If any scholar is interested to see, okay, if I run the GCMS, what would I get? If I run NMR, what would I get? So we already have such kind of information which will be available for anyone to download. So briefly, next 2-3 minutes, I will spend time on the database. So once somebody logs into it, the database takes them to a page where they gets an outline about what all information one can really get. Let me just briefly tell that you get information about medicinal plants, essential oils, their taxonomy, the profiles, their ethnobotanical uses, and also the phytochemicals which are present in them. And for pharma scholars, largely they also want, it's not only the phytochemicals which are present, they also wanted to know the ADME properties of those phytochemicals. So we also provide that information, which would be readily be available, and will help you about drug likeliness, whether they can cross the blood-brain barriers. All those features can actually be looked into, and also they map down to the biosynthetic pathways. So if one can click on the browse, go to these medicinal plants or aromatic plants, select one of those. For example, take aconitum heterophyllum medicinal plant. Then it takes you to all the information associated with that plant information, and you'll see ethnobotanical uses. These are based on some published literature, where it is used, what kind of how people use it, which part of the plant is taken, and then what molecules are present in them. So that is where the scientific insights would actually can be correlated with the drug activities which are being showcased or being discussed in the ethnobotanical literatures. So this is one. Similarly, if you take aromatic plants, which we ran in-house, and we get these multi-analytical platform profiles. For example, if I click on these profiles, I can find, say, this kind of spectra are there. I can click on one phytochemical here. For example, there is a phytochemical named as patchouli alcohol. We can see whether they are having what is the pharmacokinetic properties on them, whether they inhibit or they don't. Okay, lipophilicity, their medicinal chemistry, their drug likeliness, the solubility. So all this information, and these are being derived using Swiss ADME tool, which is freely available online. One can actually try to see, based on phytochemical molecules or any drug molecules, what is the ADMET properties of it. So overall, if you look into the features, also you can go and look into the pathways. This rhododendron, as already mentioned, we do have the pathways of those, and this is the team which who developed this. Shagun, my PhD scholar, has largely contributed to the development of this database, along with the efforts of my other scholars who have put up a lot of effort. One scholar is from pharma background. So the major takeaways of this particular part is we established a Himalayan medicinal aromatic plant phytochemical database, which is specific to the Himalayan region. About 2,500 phytochemicals profiled from 78 maps, medicinal and aromatic plants, including threatened industry relevant. Rhododendron and essential oil phytochemicals, ADME and biosynthetic pathways are readily accessible. And the phytochemical profiles and the ethnobotanical uses are other valuable information which would be available for anybody to access it. So the manuscript, once we submit it, the database will be released as well. Now, the second question is, how do we now tap into this database? Can you tap into the database and try to discover any therapeutic potential of these 2000 molecules against your targeted disease? Yes, it is possible. Let me give you a brief example and our own experience. So this work, when it was started in 2019 and 2020, the COVID time, we thought, why don't we look against the COVID, as many other scientists in India and abroad have been trying. We also thought, let us dock our phytochemical library against this. By the time that library was being developed, so my scholar painstakingly learned the tools and techniques associated with docking, molecular dynamic simulations and other stuff. And the two major targets, one is like, say, ACE2 receptor which is present on the human cell surface, and another one is the MPRO, which is the main protease which is present in the virus. So these two proteins, against these we started looking for the potential inhibitory ability of the phytochemicals. And guess what? These two molecules, the ACE2 and MPRO of SARS-CoV-2, their structures were available. So on docking with these small molecules, there are computational tools readily available. Any scholar can actually try it, can do it. Nowadays, there are online servers as well, very easy to do. But little bit of learning in relation to force fields, some of the techniques associated with them has to be studied for the fundamentals. And we figured out essential oil components, largely essential oil components. If we do, the docking scores are not very promising. Now that the more the red, the higher the docking ability. So we figured out that if we take some of these molecules which are coming from the plant extracts, especially the rhododendron, rather than the oils, we find that they are exhibiting better docking ability to the targets of ACE2 and MPRO to large extent. So that is a very good thing for us. We thought, like, okay, oils are not working fine. The flower extracts are probably working, because docking doesn't guarantee that in vitro they would be in vitro or in vivo they would be having similar efficacy. So what we thought at that time, we contacted ICGB, some scientists there, wherein we wanted to do two things. One was to do the molecular dynamic simulations, and number two is to do some experiments, inhibitory experiments on the virus. So on doing this, we figured out that they do have stable interactions against the targets, both the ACE2 as well as the stable interactions, giving good binding affinity of our molecules, especially the caffeol quinic acid, or the chlorogenic acid. And also, against Empro also, we got some decent binding affinity, but not very high. But we thought like maybe there is still some activity which is possible. It is better than all the essential oils. So once we sent these extracts, which are freshly prepared, and we sent it lyophilized, and we sent the samples to ICGB. Then Dr. Sujata, Sunil, and the team helped us to really carry some of the antiviral studies. Monkey kidney cells, the Vero E6 cells, if you take them and infect with SARS-CoV-2, then you see only virus, the viral load will be very high. Of course, the viral will replicate inside these host cells, and their numbers would be very, very high. The titer of the virus would be very high. Whereas if you do a dose dependent, if you provide the dose wise concentration of these arboreum or the rhododendron extracts, flower extracts were provided in a dose dependent manner, then you see as the dose increases, the amount of viruses which are being eventually produced or replicated inside the host cell, they would reduce. So that gave us an idea about IC50 values in relation to the rhododendron extracts. Now we know these rhododendron extracts phytochemicals are largely quinic acid derivatives. So, and the docking studies also helped us to really figure out like there is a stable interaction of many of those molecules. Then what we thought, we extended our docking studies against all the 22, at least I think 22, just let me check. Yeah, we docked against 22 target proteins, and then we figured out these quinic acid derivatives, they indeed have very good docking potential against several of the targets. So our statement in this regard is that there would be multiple phytochemicals which might be acting against multiple protein targets, and that's why we rated this particular rhododendron flower petals as promising antivirals. So this led, to ask the question whether they have broad spectrum antiviral ability. So further studies we carried again with the same group. I acknowledge again Dr. Sujata, Sunil, and her team, wherein they tested against the chickpea. Although the compared to the SARS-CoV-2, the inhibition or the was comparatively lower, but still the rhododendron extract seemed to be having some antiviral ability there as well. I'll keep this short, and then this is like chickpea, chlorogenic acid, and quinic acid. Now what we did is, now we started taking the individual components from this flower and as well as from the standards, and started working on the IC50 values of the individual components, and we are seeing whether they are toxic to the cells at the concentrations where the viral inhibition is happening, and those kind of studies. Now we are at a stage where we wanted to do the mechanistic insights within the cellular systems, how it is working. See, that's the ongoing work, and interestingly, these quinic acid derivatives, they don't show any effect on microbial systems, which is good actually. That means if on human consumption, your gut and others wouldn't be influenced. We figured out that whether E. coli, gram positive or gram negative microbes, or even acid fast strains like Mycobacterium smegmatis, they don't show any inhibitions. Only at very, very high concentrations they show some inhibition, but that much concentration nobody really consumes as a drug candidate also. So we figured out like, see, these quinic acid derivatives, they doesn't show any effect on microbial systems either, which is good, but they have got effect as antivirals. So the major takeaways from here is that the rhododendron petal phytochemicals, which were already, we did the metabolomics, we know the profiles, we know the molecules, they exhibited promising binding affinities against SARS-CoV-2 target proteins, that too in a dose dependent manner. And we know that largely these are quinic acid derivatives. So now using this information, we patented this. Patent is already awarded, and also we published the same information in a respectable journal. So I could encourage the scholars to look into those. So now that the library is there, we found antiviral. One more scholar, PhD scholar, who is currently working, came about, and he wanted to work on the MDR strain and wanted to use the same phytochemical library to look for molecules or potential candidates with antimicrobial resistance, which are becoming MDR, multi-drug resistant. And interestingly, our group has currently discovered a pathway. I'm not going to name it. Discovered a pathway which is common to at least 19 pathogens infecting humans. So some of those pathogens happens to be Salmonella typhi, Shigella species, Staphylococcus aureus, Helicobacter pylori. They cause, some of them cause, like said, Salmonella typhi for typhoid fever, H. pylori, the very high relevance related to ulcers and gastric cancer. So now that we discovered a pathway and we know the target, protein target, my scholars are currently putting a lot of efforts in order to do the screening, and then we wanted to test against the pathway to inhibit it. Interestingly, the pathway which is present in these pathogens is absent in humans. That means that could be a potential drug candidate, drug target. So the screening process involves this way, wherein from the molecule, from the microbes where we are targeting, we do a lot of metabolic systems biology studies, metabolomics studies, and we eventually identify the key bottlenecks of the pathways, and then we wanted then we to the targets using AlphaFold. If the protein structure is available in PDB, we take it. If not available, we use AlphaFold, the AI ML based platform, to predict the structure. Once we predict the structure, we do some validations, and meanwhile we use our HMAP square database to bring those small molecules and try to dock against some of these protein targets. So on doing that, we find we get initial screening and leads. Now the leads goes into the lab for further antimicrobial or antibacterial studies. So that is how currently we are using so many essential oils directly, where we are trying to find their antibacterial activity, antibiofilm activity, and we anticipate them to be potential new antimicrobial molecules. We are doing it against Mycobacterium at the moment, and as well as other microbes which I just mentioned. So, for example, in one of the candidates from the HMAP square database, we found that it has the ability, very good ability, to actually inhibit the biofilm formation, both on the pre-exposure as well as post-exposure. I mean, before the biofilm formation, if you expose, no biofilm is formed. If biofilm is formed and you expose the biofilms as well as the microbe with one of our essential oil as well as the phytochemical mixture, which is synthetically designed, we see that it can disrupt the biofilm at the moment, which will help to develop novel antimicrobial or antibacterial components and lead to the drug discovery. So that brings us to the last case study, which I'm very excited about as a plant biologist myself. If you look into the aspect, there is something, a new terminology, or pretty much I think it's there for a while, is called is functional foods. That means food itself can act as a therapeutic component, having some potential to overcome some diseases, or at least protect us from some diseases. And it is also known that there is a hidden hunger wherein the plants which we are consuming might be devoid of the essential micronutrients in some cases. We consume a lot, or the plants are being engineered to produce a lot of biomass, but they might not be having the key nutrient components in them. So our hunger is taken care of, but our actual body requirements are not taken care of. So we call them as, like, say, hidden hunger. So in order to address that, we also use nutri-metabolomics, nutritional and metabolite profiling, nutri-metabolomics of some microgreens. I think if you search in the Google microgreens, you'll see in India many people are producing this right now. So if you have a small room and racks and some light with LEDs and others, one can really produce a lot of microgreens. These are, like, say, seeds which are germinated until they get to 2-leaf stage. And it is known that all the seed minerals and other nutrients, they are released in such a way that they become more bioavailable in the microgreens. Currently, we have an industry partner as well, with whom we are working on these functional foods to develop the next generation smart foods from this. So from when I did on the brassica microgreens, that's one example, but we are doing it on almost 20 odd microgreen varieties. So I'm showing you 4 here. On doing with the 4, we figured out they do have very good amount of polyphenols, antioxidant ability, and these can serve as a serve and contribute to the nutrition. Let me go a bit detailed into that. For example, if one consumes 10 grams by dry weight of brassicaceae microgreens, you may get like iron about 51 percentage. If you consume 10-gram dry weight equivalent of microgreens, you get 51 percentage of the required daily allowance of iron is taken care of. Manganese of 29 percentage. Of course, zinc in these microgreens, the zinc is only about covering about 8 percentage. But then one can really formulate the microgreens which can be enriched separately with zinc as well. So the point I'm trying to make here is one can develop functional foods in such a way that it takes care of the human required daily allowance. So that is possible. Similarly, we did like again metabolomics, the metabolite profiling. We figured out what kind of molecules are present in them, sugars, amino acids, organic acids, amines, fatty acids, other phytochemicals, largely, secondary metabolites or primary metabolites. And we figured out they do have very distinct metabolic profiles. And there are like significant levels of some molecules which are very high. For example, there is a polyphenol, which is very high in brassicaceae micrograms. Now, and also brassica being oilseed family, we found them, they do have very good amount of essential or beneficial lipids as well. Like say some of them as fatty acid like linoleic acid, oleic acid, or stearic acid. And we wanted to further take this forward to see how these can act as a functional food, their bioavailability and so on. So the key findings, ignore the chapter 4 in this case. I took it from a PhD scholar's slides actually. The key findings from the mustard as functional food, you can look into they have got promising nutritional and antioxidant abilities. They have got promising level of some of these molecules, which are, these micronutrients, macronutrients, which are essential for us. And then a range of phytochemicals on consuming them are available. Many of them are beneficial to the body, essential ones, essential fatty acids, and so on. The fatty acid profiling has given us, some of the essential fatty acids for our body, which are really important. And largely, we feel that many microgreens, they will provide at least 20% to 50% of RDA by consuming very little amount of these microgreens, rather than having so much of anything. You can have little but very high quality. So with this, we mentioned that the functional foods will have promising potential to overcome hidden hunger. So further reference can be referred so that you can find out more details associated with this particular work. So let me briefly remind you and also give an overall conclusion of the talk. The first one is using metabolomics, metabolite profiling, one can do the comprehensive phytochemical profiling of MAPS, which we did in this case. And this can provide us an idea about the diversity of these bioactive molecules, potential bioactive molecules, and future drug candidates. And they also capture the diversity, giving us a lot of fundamental knowledge about how these plants are making them. So if we identify them to be of any value, we know the sources and how we can really scale these up to produce the required phytochemicals. Second, we developed a HMAP square database, which will serve as a valuable repository of medicinal and aromatic plants and phytochemicals, especially those from the Himalayan terrain. Those 22%, out of which many are rare and endangered, we really tapped into them, including the industrially relevant essential oils, and those would be very valuable resource. Then the third one is we have demonstrated that some of the phytochemicals, especially from the rhododendron, they are having antiviral potential. I don't call them yet broad spectrum, but we do hypothesize them to be having broad spectrum antivirals. That is the reason why we patented this particular formulations based on which rhododendron arboreum petal extracts can be one of the key components in the antiviral, as well as the molecules present in them can be the potential antiviral candidates. So more studies are ongoing in that direction. And finally, the microgreens as functional foods, which is largely based on the nutri-metabolomics. Metabolomics and nutri we integrate here in order to come with functional foods, which will help us in alleviating the hidden hunger part. The future directions, if I were to speak, one can do the discovery of the dark metabolome, because we acquired so many spectra, so much data is lying, raw data is available. The dark metabolome, unknown peaks, whatever we are naming, or the peaks are being identified, they are largely in relation to the available libraries. But one can further dig into it to see, are there dark metabolome which with unknown peaks? Can we capture them? That information could be vital. Number two, and though could be biotic. Number two, the HMAP square, we can go ahead and use the same library for identify against several other diseases. One scholar at one point of time was looking for anti-mosquito, the larval inhibition ability of the phytochemical, so that the mosquito menace can be controlled. If we can really come out with those candidates which will stop this larva to germinate, or some to grow. So that is one thing which one scholar was looking at. Like that, any disease of your interest, as long as targets are available, one can tap into this phytochemical library. Third, we have shown we wanted to use multi-omics approaches like transcriptome, metabolomics, fluxomics to derive the mechanism of action. It's not sufficient nowadays to just say it works. Okay, we demonstrate it works, but we wanted to know the mechanism of action. So that's where our energy is being put into. And finally, the therapeutic functional foods, you can see very colorful. These happens to be different light wavelengths. So one PhD scholar is currently looking into the effect of different light wavelengths on the modulation of the metabolites within this functional food microgreens. We figured out, if, for example, if you expose the microgreens to blue light after 8 days of growth, we see there is a several fold increase of essential fatty acids like omega-3 or omega-6. So that, or you can give a combination of blue plus far red, or again blue plus far red, so that you get a lot amount of these essential fatty acids which could be elevated. Could be a game changer and could lead us to designer microgreens with enrichment of targeted molecules. And that is where the nutri-metabolomics is really powerful and can be tagged with therapeutic abilities. So with that, I would acknowledge, of course, the support I received from IIT Mandi, my school, Biosciences and Bioengineering, my group, FluxMets is our metabolic systems biology laboratory, my collaborators across various institutes, ICGB. Although it's not here, but I should really acknowledge ICGB collaborators. The funding sources, pretty much all the funding sources. Also, I would like to acknowledge especially the funding coming from the Ministry of Education, supporting my scholars, MHRD, Imprint, Spark, SERB, Himalaya Wellness. Also, we have a project from one of the HPSMB from the Himachal Pradesh State Agriculture Marketing Board. And largely, I would also acknowledge my PhD scholars. 7 of them have currently submitted or graduated. And also acknowledge all my scholars, PhD, MTech, postdoc, and women scientists, fellow scientists whom we hosted, interns who come and work and go back and excel. I would say many of them are in industry, academia, and so on. So thank you very much. So I'll be happy to take any questions you may have.
Thank you, sir, for the insightful talk. We'll now take a couple of questions from the audience. If anyone wish to ask any question to sir, please, the platform is yours to ask.
I would stop sharing. Yeah.
Anyone wish to ask anything to sir? The platform is yours. Okay, sir. As we conclude this engaging plenary session, I do like to extend heartfelt thanks to our speaker, Dr. Masakapalli, sir. Thank you, sir, for joining us and delivering a keynote address of e-National Conference on behalf of my institute, IPER, organizing committee members, and on my own behalf. I express my deep sense of gratitude for sharing your valuable ideas and knowledge with us, sir. Thank you. Thank you very much, sir.
Thank you, Dr. Manisha. It's a pleasure indeed, and all the very best ahead as well for the conference. I've seen that some of my scholars also joined here. So yeah, thank you very much.
Thank you, sir. Thank you very much, sir.
Yeah.
Thank you, sir. Thank you, Dr. Shyamkumar Maskapalli, sir. Thank you.
Thank you. It's a pleasure. All the very best.
Yeah.
Thank you, sir. Dear delegates, the next session will be at 2 p.m. sharp. The next session is at 2 p.m. sharp. Using the same Zoom link, the next session is at 2 p.m. sharp. Take note.
Thank you, madam.